Oh, all right. Let's get let's get into it. So last week or the week before, whenever I last put out a video, I've not been good about it lately, so I apologize for that. But Tri4 gets busy around midterms, and I know you guys have been busier than all get out with Tri3. So you guys are doing good so far, making it through. I'm hearing a lot of good things from you guys, so we're just gonna keep on cracking away. But in my last video, I know we talked about beta oxidation of uh, fatty acids, I believe. We talked about unsaturated fatty acids, ketones bodies, how it can put your body into ketoacidosis just by using up those bicarbonate ions in the blood plasma. So if you're kind of unfamiliar with that, just go back and watch the last video, get re-upped on that. And I know a lot of you guys go to Annalise as well, and she does a phenomenal job. So there's a lot of great sources. So we're just going to keep on cracking away. And honestly, today we do not have a lot to cover. It seems both Biochem 1 and Biochem 2, Goot has been he's been cruising through a lot of material. So usually this red line right here is the cutoff for exam 2 content, but I know he has been talking about some of these concepts of the regulation of gluconeogenesis. So I am just going to go ahead and cover that as well, just so we're not caught off guard with anything. Um, but realistically, the material that we're going to cover here is not too terrible. And again, with the practice problems, if you bounce between this and the practice problems, you will know everything you need to know. And I'll be honest with you, towards when Tri3 started getting busy, I didn't put as much time into studying for this class. So like a lot of this information, I know the superficial knowledge, but nothing, nothing super in depth. So we are going to be talking about proteins, and similar to how we talked about carbohydrate digestion earlier, it might have been lipid digestion. This is just going to involve proteins, and it's very similar concepts, nothing too bad. So with proteins, we are going to be ingesting long-chain polypeptides, and we're going to go through two different stages to break them down into dipeptides, tripeptides, and eventually individual amino acids. Biggest key points for this section is to know what area, whether it's the stomach or intestinal lumen, breaks down these proteins into specific components. So as I just said, digestion occurs in the stomach and intestinal lumen with different enzymes. And in the stomach, all we really need to worry about are pepsin and pepsinogen. So pepsin in our stomach is going to remain in an inactive form known as pepsinogen. And basically how pepsinogen becomes activated to turn it into pepsin, which is the active form, HCl and the low pH of the gastric fluid is going to activate pepsinogen to transform into or become activated and become pepsinogen. And what we need to know is it digests the dietary protein strictly into polypeptide chains. So we're taking a large tertiary quaternary structure, breaking it down, kind of denaturing it into just a polypeptide chain. Now, we do not have individual amino acids yet because we still have those peptide bonds in between each individual amino acid. And as specified in the red, let's see if my laser pointer works this time. As we can see right here, it cannot digest oligo or amino acids. And the only amino acids that can be separated by pepsin are aromatic amino acids. But when we go from the stomach into the intestinal lumen, where these pancreatic enzymes start to take role, that is where we can start breaking down some of these polypeptide chains into individual amino acids. So we have two kinds of pancreatic enzymes that we need to worry about. We need to worry about endopeptidases and exopeptidases. So endopeptidases are trypsin, uh, chymotrypsin, I don't know, and elastase. And then for exopeptidases, we're just going to be only worrying about carboxypeptidase. So as I said before, small intestines produce enzymes to finish the digestion of peptides, so that only individual amino acids remain. So aminopeptidases, tripeptidases, da 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 da. But we need to worry about those enzymes I just talked about right here. So endopeptidases, which include trypsin, chymotrypsin, and elastase, these enzymes hydrolyze peptide bonds in the middle of the chain. So and I know you guys probably are picking up what I'm putting down, but just for the sake of it, imagine we have a little polypeptide chain right here with all these amino acids. Endopeptidases is going to come right in the middle and just start breaking the bonds starting from the middle of the chain. So I would recommend memorizing these enzymes and just being able to comprehend which ones are endopeptidases, which ones are exopeptidases. But I honestly, for the exopeptidase, I just memorized that carboxypeptidase was the longest word, and I was like, longest word goes with exopeptidases. So come with your own little trick. I did not put much effort in towards the end of this unit, and it definitely shows when I try to tutor it for the first time. So let's talk about exopeptidases now. These enzymes hydrolyze a single peptide bond at the C terminus end of a polypeptide chain. So instead of coming in and attacking the middle, we are just going to be attacking the C terminus, or whatever end the C terminus is on, and just hydrolyzing individual amino acids off this chain. So these two enzymes have just two distinct roles. And honestly, this is just strict memorization. You don't need to go super in depth with this. There's probably going to be maybe 2 to 3 questions on this concept. I've only been seeing maybe 1 or 2 per practice sheet, but it's not something to be overly concerned about. Just memorize. Pepsin is in the stomach. These pancreatic enzymes are present in the intestinal lumen, and just being able to differentiate what enzymes are endopeptidases and exopeptidases and what those specific roles do. Amino acids exit the GI through the hepatic portal vein, eventually end up in the liver, same as carbohydrates, different than fats. But yeah, for that, that's all we really need to know. So now we're going to start moving on to nitrogen and the amino acids, because amino acids help supply decent bit of nitrogen to our body, but we can only utilize it under specific circumstances. So if amino acids are to be used in gluconeogenesis or converted to fatty acids for storage, they have to be stripped of their nitrogen. We cannot have this nitrogen attached. We need to separate it first before we can start utilizing some of these amino acids. So we do have 2 methods for removing nitrogen. We have oxidative deamination, which oxidative deamination, all that really means is we're just stripping a nitrogen off here. And so glutamate and alpha-ketoglutarate are a great example. And realistically too, if you guys were at my past couple tutoring sessions, we did talk about transamination reactions and how you need to memorize that alanine goes with pyruvate. We know aspartate goes with oxaloacetate, and we know glutamate goes with alpha-ketoglutarate. So for method 1, or oxidative deamination, anytime you see glutamate, even if it's oxidative deamination or transamination, we should just kind of understand that glutamate is eventually going to possibly turn into alpha-ketoglutarate under most circumstances. And most of the questions you guys will see on this, it's pretty cut and dry when it comes to just knowing which goes with which. So, sorry, a little tangent there, but glutamate will be acted upon by the enzyme glutamate dehydrogenase, and all we're doing, again, is stripping that nitrogen or amine group off, and we'll turn into alpha-ketoglutarate there. And we do gain energy from this. And if you guys remember NADH, these guys can go through redox reactions here and give off energy. So NADH will go react with half of an oxygen to produce H2O and NADH, and we're also going to be producing some ATP, 2.5 ATP specifically for these NAD plus and NADHs. But besides that, for oxidative deamination, we really don't need to know too much. So now we get into transamination reactions, where instead of it just being stripped, we're kind of doing a transfer of amine groups here, and the main enzyme acting upon this is aminotransferase. And I really did not get too bogged down with this information here. I just used these little cues on the side. If you guys, again, if you want to go and memorize this, go and memorize it. I'm never going to say that's a bad thing to do. But transamination transfers an amine group of an amino acid and a carbonyl group of another to form 2 new molecules. So you can see that here we have an alpha keto acid and an alpha amino acid going off like that. And we'll end up with 2 different molecules, 2 new molecules here. And this was prevalent in the malate shuttle that we talked about a decent bit back. Let's see if I can find it right here. Right here, transamination reaction. We can see it's glutamate, oxaloacetate, transaminase. and we can see oxaloacetate here. Um, Oxaloacetate and glutamate kind of go through a transamination reaction here, and we can see that oxaloacetate will transaminate into aspartate and glutamate will transaminate into alpha ketoglutarate. So that's kind of a functioning aspect of a real example that you guys are going to be looking at here, and I believe I talked about transamination in my previous videos, but this is a good practical view of it. So back down to where we were. So we should know that, again, alanine goes with pyruvate, aspartate can transaminate into oxaloacetate, and glutamate can transaminate into alpha-ketoglutarate. And what I would recommend knowing for these are these carbon counts right here. So pyruvate and alanine are 3 carbon molecules, so there will be some practice problems you might see where he gives you the molecular structure and gives you 2 molecular structures, an arrow, a blank space, and another molecular structure, and he's like, hey, in this blank space here, what would be the molecule that's present here? So the best way to go about that is look at the beginning structure. And this is really hard without drawing it out. I should have been a little bit more prepared. But basically, all you want to do is count the carbons in each molecule. And some of them might have carbon rings. Do not worry about the rings. Just worry about strictly the carbons in the structure. So let's say you have a structure with, actually, you know what? Screw it. Screw it. Let's find a blank space here. Let's say, and I'm not going to follow normal rules here. So let's say you see this structure and he's like, oh, bust that. Like, let's see, HC hydrocarbon structure there, and you also might see one over here. CH, blah, blah, blah. So like, let's say Goot wants to know what structure is going to go right there, and he's going to be asking about a transamination reaction. So what I always do is I look at the structure, count 1, 2, 3, 4. So I know I have a 4 carbon structure here, and you can see right here we have 3 carbon and another, let me throw another carbon in there, another 3 carbon structure right here, and we can see that this structure and this structure are going to be the same. So he's really referencing this structure and what's going to be the name of it in the second half of the equation. So if we see 4 carbons here, we should look down and just kind of memorize this chart right here. We can see, okay, well, aspartate and oxaloacetate are going to fit that 4 carbon structure, and they'll just be a bank, possibly with all these different, you'll have probably like pyruvate, alanine, alpha-ketoglutarate, glutamate all in there, but you'll only see one of the correct answers, which either could be aspartate or oxaloacetate, depending on the style of the question. But the key component here is just to count the carbons and just match up what it's going to be going with. So in this case, we really want to be looking for either aspartate or oxaloacetate as the correct answer, since we know it's referencing a 4 carbon molecular structure. Very long-winded example, but I saw that the other day and I was like, that is just a very good tidbit of information to know. So yeah, honestly, just again, memorize what phases go right there, and it also, again, works for deamination as well, as we can see right here with glutamate going to alpha-ketoglutarate. And this example will pop up in the urea cycle, as I said, which we'll get to in a second. Oh man, I'm talking and just not breathing. I'm running out of breath. I'm getting lightheaded. Biochemistry gets me excited, man. All right. So what we do need to know is the B vitamin that does go with these transamination reactions, and it's going to be referred to as pyridoxal P, which is a B vitamin, which should be B vitamin 6. Yep, or you might see it as pyridoxine. Oh, where was that B vitamin chart at? Referred to as pyridoxine. So he will either have pyridoxal P or pyridoxine as an answer choice for vitamin B6. So just be familiar with being able to bounce back and forth. And we're going to be getting into amino acids here in a second. We're pretty much done with these transamination reactions. We know that pyridoxal P, which is vitamin B6, goes along with these transamination reactions, and this section is pretty much good for what we need to know at this moment. So now we're going to start talking about some different amino acids, and I personally did not get super in depth with this. My main components here where I memorized that leucine and lysine were ketogenic amino acids. And what ketogenic amino acids stand for is these amino acids are strictly going to be used for the formation of ketone bodies. Anything that is used for the formation of ketone bodies cannot be used for gluconeogenesis. So if you, like even right here, you can see that leucine is a part of acetyl-CoA, and it will go to isoacetyl-CoA, because if we remember from ketone body formation, we need two acetyl-CoAs to begin that process. So whether these amino acids go to acetyl-CoA or isoacetyl-CoA, we know that they cannot be used for gluconeogenesis, and that's why there's a pretty big distinction between leucine and lysine. And those are the main two I remembered. And if I did not see those two, I just assumed all these other amino acids were going to be used for gluconeogenesis. And there is some overlap if you guys do want to memorize isoleucine and tryptophan and tyrosine and tryptophan. If you guys want to memorize those, go right ahead. But I strictly just looked out for leucine and lysine. And another big component I want you guys to understand too is besides these ketogenic amino acids right here, all these other amino acids around this chart are being inserted at some point on the TCA cycle. The TCA cycle is one of the cycles that takes advantage and uses these amino acids to either go through gluconeogenesis, supply some of these functions and everything like that. So those couple of distinctions and tidbits of information right there are going to get you a decent way. But again, please practice on your own. You're going to learn it way better than me just spitting all this information at you. So let's just go right here. I'll come back up to that PEP or phosphopenylpyruvate in a second. So amino acids can be used to produce glucose and ketone bodies from their actions in the TCA cycle. Glucogenic amino acids are the ones that can form alpha keto acids that are converted to PEP for gluconeogenesis, and then ketogenic amino acids are ones that can form S-acetyl-CoA, which form ketone bodies, basically what I just talked about. Let's look at PEP, which is phosphopenylpyruvate. PEP right here, and if we remember, phosphopenylpyruvate, if we remember from the glycolytic pathway, I apologize for my handwriting. PEP was the one molecule right before the formation of pyruvate. And if we remember, we did produce an ATP here at this instance to put pyruvate into, or convert PEP to pyruvate. We made an ATP, and pyruvate could either go down here to the TCA cycle in an aerobic condition, or we could go to lactate in an anaerobic condition. But I just wanted you guys to understand where PEP is located in reference to the glycolytic pathway. And PEP is a precursor to gluconeogenesis, and certain amino acids are also precursors. And again, as we just covered, these are glucogenic amino acids. And basically, we can go around this TCA cycle, get to oxaloacetate, and oxaloacetate will turn into eventually PEP. And then from there, realistically, we are just going to shoot back up the glycolytic pathway to go all the way back to glucose. Now, in the liver, we go all the way back to glucose, but there might be a question where he's like, outside of liver cells, where is going to be the stopping point at? And as we know, the liver is the only organ in our body as of per this course that contains glucose 6-phosphatase. And if you guys remember, glucose 6-phosphatase converts G6P into glucose. Now, if he's asking about gluconeogenesis in any cells outside of the liver, that glucose 6-phosphatase will not be present anywhere. So instead of glucose being our endpoint instead will be G6P. And then G6P can be used to go into glycogen for storage in our muscles and everything like that. But having that understanding of how far some of these processes go, depending on the cell you're in, is a main key point that we need to know. Right here, good point to know. Acetyl-CoA cannot be a precursor for gluconeogenesis since it needs to react with oxaloacetate to begin the TCA cycle. And we need oxaloacetate to begin or help with gluconeogenesis too. So if we are using oxaloacetate to react with acetyl-CoA, we are taking away from our oxaloacetate supplies, which we need right here to begin gluconeogenesis. So if we don't have that, gluconeogenesis is going to be acting in a less efficient manner and just isn't proper for our body to do. So if there's an option to where talking about, or if there's a question talking about, hey, which of these components can begin gluconeogenesis, and you see acetyl-CoA there, do not pick that, because acetyl-CoA needs to react with oxaloacetate, and we just do not want that for gluconeogenesis. If there's any option already in the TCA cycle, like for example, succinyl-CoA, fumarate, malate, any of those options, yes, they can begin gluconeogenesis because they are already in the TCA cycle and are not using up oxaloacetate supplies. But anything outside of the TCA cycle, like over in this realm, cannot be used to begin gluconeogenesis. And again, a lot of these reactions are occurring during starvation. We've used up all of our carbohydrate stores, and now we need to rely on protein breakdown, and that is no bueno if you're trying to get big in the gym with all your protein. All right, that's enough talking about that. I'm kind of sick of amino acids now. Let's keep on moving. Phenylalanine, yeah, it's pretty cool. It's ketogenic and glucogenic. It's essential amino acid, which means that we need to ingest it from outside dietary sources. We cannot produce it in our body, therefore we must ingest it. Under normal conditions, phenylalanine gets oxidized to tyrosine, which is a normal functioning process in our body. And if you guys remember all the way back from, I think it was exam 1 content when we talked about the pentose pathway, we know that the pentose pathway produces NADPHs and NAD pluses. And what's really cool about the pentose pathway is, on top of helping out with some RNA functions, everything like that, we produce some of these metabolites like NADPH plus to go towards anabolic pathways, which means building larger structures. And this is a perfect example of NADPHs coming back because we're taking phenylalanine and we're creating a new structure or a larger macromolecule called tyrosine. Tyrosine is an important intermediate to the production of catecholamines and acetoacetate and fumarate. Catecholamines being epinephrine and some of these examples down here. So we know we have catecholamines right here, we have fumarate right here, and honestly, if tyrosine is further broken down through tyrosinase, I think that's how you pronounce it, we can also produce melanin. One of the main enzymes I want you guys to know is phenylalanine hydroxylase, which is the enzyme that breaks phenylalanine or converts phenylalanine into tyrosine. Now, when we talk about some disorders, this is going to be the main enzyme that is malfunctioning. So, leading right into that, when phenylalanine hydroxylase is not present, phenylalanine is converted into phenylpyruvate. And this is a ketone body that can lead to metabolic acidosis and mental retardation. So it is a no bueno, and it is a very bad thing if we have phenylalanine hydroxylase, or if we do not have phenylalanine hydroxylase. How I always remembered it for just taking an exam, I just thought right back to our I'm at a loss for words for a second. Kind of the same concept for transamination reactions. If you talked about phenylalanine being converted into phenylpyruvate, I was just like, oh, like alanine goes to pyruvate. I know phenylpyruvate is bad and it can lead to all this ketone body formation, bad stuff. And I was like, okay, so phenylpyruvate, bad stuff. Tyrosine, good stuff. Another thing we should know is phenyl or PKU. Oh man. PKU. Oh, sorry. I am all over the place today. Phenylketonuria is an autosomal recessive disorder which causes a lack of phenylalanine hydroxylase, which can lead to the formation of phenylpyruvate and then down to phenylactate. And if someone does have this condition where they do not have enough of phenylalanine hydroxylase, the best way to bypass it is to just ingest high amounts of tyrosine to keep the production of melanin, keep the production of L-DOPA, catecholamines, acetoacetate, fumarate, to keep all these functioning running. So that's just kind of a nice bypass that people that do have this autosomal recessive disorder, this is a good way for them to get around it and not be overly affected by it. Hey, what? All right. Sorry for that. All right. we're already getting close to the end. I mean, we're kind of cranking through this. So the urea cycle. I know I name dropped this structure earlier when we were talking about oxidative deamination, and now we're here. So the urea cycle is kind of how we strip nitrogens off some of these proteins or amino acids, and it's also kind of how we excrete these nitrogens from our body. And then we can kind of relate this back to biochemistry 1 when we were talking about primary alkali deficiencies and like acidosis, alkalosis. If you remember, with acidosis, one of the main components that we excreted was NH4, and that was an acidic component. The acidic component of this really doesn't apply here, but this is just kind of a downstream effect, as we can see here with urea. Eventually, when we get to the end step, it goes blood, kidney, urine, kind of how our compensation mechanism in our body. But let's start from the beginning. So the urea cycle starts with glutamate, and glutamate goes through an oxidative deamination reaction, which, as we remember, is the stripping of the NH4, which is this right here. Whoop. Stripping of the NH3, which eventually would turn into NH4, da 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 da. So, sorry, urea is produced in the liver, and this whole mechanism is kind of happening in the liver. What I want you guys to know is that in here, this is all inside the mitochondrial matrix right here, this nice little ring. And then a lot of this on the outside is all happening in the cytosol until eventually these products leave and go off into their respective areas. Now, we can also see that we do have an ATP count right here. And personally, I like using this chart, but we'll reference this chart as well to talk about the ATP counts. So I'm just going to walk through here. We'll cover the ATP counts up here in a second. So what I really want us to know is we're just going to be starting from here, which is step number 1, which is going to be stripping that nitrogen off of glutamate through oxidative deamination. So that NH4 is going to enter into the mitochondrial matrix, and it's just going to have a couple reactions. It's going to combine with a couple things. We're going to use some ATP here. And what I want us to know is it will eventually become attached to this carbamoyl phosphate. I just called it C-phosphate. But what we need to know for C-phosphate is that it is kind of just the carrier for this amine group, and I want you guys to reference it as the carrier. This C-phosphate is going to latch onto this amine group or this nitrogen group, carry it down through the mitochondrial matrix until we have the presence of L-ornithine present in the mitochondrial matrix. And as soon as L-ornithine is present in the mitochondrial matrix, this C-phosphate is going to drop off this nitrogen onto it. So we know for this little step inside the mitochondrial matrix, C-phosphate is going to be one of the first components that inserts nitrogen into the urea cycle. So we're going to be plus NH4 right here. And as soon as that nitrogen group is attached to L or not, L ornithine. I can't even pronounce it. I don't even care anymore. Okay, once the nitrogen gets attached to that, it's going to leave the mitochondrial matrix and enter the cytosol as L-citrulline. So as we can see, L-citrulline has the one nitrogen group right there. It's changed forms from this little reaction right here. And we're going to get the addition of one more nitrogen group from L-aspartate right here. So the two components that are going to insert nitrogen into the urea cycle are going to be C-phosphate right here, and then L-aspartate right here. These two components will insert the nitrogen into the urea cycle. Now, L-citrulline is going to go through a reaction with L-aspartate, and we're going to drop off another nitrogen, and then it's going to turn into arganosuccinate up here. We're going to lose a structure right here and form fumarate, which will go through the TCA, turn into malate, oxaloacetate, then eventually into alpha-ketoglutarate. This is kind of describing the malate shuttle a little bit right there. And then once this little component leaves right here, arganosuccinate is going to turn into L-arginine. And then eventually up here, we're going to turn into urea, and that urea is going to be excreted downstream. So now that we kind of have a basic understanding of this stuff and this little chart, we'll just run through it right here to understand how these ATP counts work. So we can see glutamate right here goes through glutamate dehydrogenase, which means we're going to be losing a hydrogen, which means glutamate is becoming oxidized and NAD plus is becoming reduced. And we can see right here that with this little redox reaction right here, we produce 2.5 ATP. Now, when we have the NH3 and we need to attach it to C-phosphate, we need to use 2 ATP to do that. So we are plus 2.5 right here. Now we are minus 2 right here. And we can see we also have the addition of a carbon from that CO2. And once that C-phosphate gets that nitrogen, it is going to come down here and react with ornithine to produce citrulline, as we said. So we know that this nitrogen right here is being transferred over into ornithine, and eventually we're going to form citrulline right here. Then citrulline again is going to pick up another nitrogen group from aspartate right here. We're going to turn into arganosuccinate, and then down here we talk about fumarate, and eventually we'll come back down here. And when we get to malate, fumarate will eventually turn into malate, and for malate to turn into oxaloacetate, again, we need to take away a hydrogen, which is going to be dehydrogenase, and with us taking away a hydrogen, we set up for another redox reaction right here. So again, we're another plus 2.5 right here from malate going to oxaloacetate, and oxaloacetate will go off and do its own thing, or it'll go through an aminotransferase reaction and we'll regain our aspartate right there. These little smaller steps really are not as important, but what I want us to focus on were these ATP counts within the urea cycle. And basically all we need to know is in the urea cycle, we produce 5 ATP, which is 2.5 right there, 5, but we also use 2 ATP at this intermediate step. So in all reality, we produce 5, but our net count is 3 ATP. That was a lot. Yeah, I think that's really it. Honestly, I don't care. I really don't. Well, no, that sounded bad. I do care, but I think we covered enough for what we need to know. This stuff, go through it on your own time. I really did not look at it too much. I don't think I even looked at this section once when I was studying for it. Uric acid is not urea. All right, 2 pages left. Yeah, 2 pages left. We got this. So now we're going to do a quick overview of gluconeogenesis. And if you guys remember, gluconeogenesis is the creation of glucose or G6P downstream from pre-existing components or molecules within our own body. And a lot of these components begin within or around the TCA cycle. Yeah, so this page right here is talking about pyruvate specifically, because pyruvate is also involved in the act of gluconeogenesis. And also, I'm going to give a shout out to the sparkling water Waterloos. If you guys hear me pausing and gulping on something, it's these Waterloos, these sparkling waters. Phenomenal. So let's talk about the different pathways of pyruvate here. So if you guys remember from exam 1 content, we have lactate dehydrogenase right here. We have lactate. Lactate can go through lactate dehydrogenase and turn back into pyruvate. Then the pyruvate can go through the PDH system and eventually turn into acetyl-CoA. And in conditions to where we have a good amount of carbohydrates in our body, we're able to produce energy from carbohydrates, and we have a good supply. We're going to go through basically this kind of side of things right here, because we're breaking down and catabolizing these glucose molecules to produce ATP and produce energy through the glycolytic pathways, both aerobic and anaerobic. But what happens if we don't? What happens if we run out of these carbohydrate storages and we need to use some of these inner components? So this is where this half is going to start coming into play here. So we can get pyruvate is just going to be a nice central molecule, and we can create pyruvate from amino acids, or like transamination reactions, how we just talked about, because alanine and pyruvate kind of go back and forth with transamination reactions. So if we have alanine, we can put it through alanine transaminase and turn it into pyruvate. And then we can also carboxylate pyruvate down into oxaloacetate, which means we just add a carbon on, because if we remember, pyruvate is a 3-carbon structure. So if we throw one more carbon onto that structure, we can convert it into oxaloacetate. And so with pyruvate carboxylase, we are going to be using 1 ATP, and we are also going to be throwing on a CO2 to pyruvate, and our result or molecule is going to be oxaloacetate. And oxaloacetate is going to kind of be one of our exit points to begin gluconeogenesis or to help start it. And it is going to use the malate shuttle to help produce it, and we're just going to keep on rolling from there. So realistically, this side, glycolysis, this side, gluconeogenesis. All right, let's navigate. Yeah, let's navigate this chart right here. So if we want to know where this little chart thing happens right here, we're going to be looking at this beginning system right here. And I'm gonna ignore a lot of this stuff over here and just focus on everything on the left. So again, we have pyruvate right here. Pyruvate gets acted on by pyruvate carboxylase, which means we use an ATP and we also add a CO2 onto it to form oxaloacetate. And oxaloacetate, if we remember from the malate shuttle, it cannot cross the mitochondrial membrane. We need to convert it into malate to get it across the mitochondrial membrane, and then we can convert it back into oxaloacetate on the other side. So oxaloacetate, and if we, actually I want to do a little drawing. If we remember malate, it goes right here, we turn into oxaloacetate. And if we remember, if we want to turn malate into oxaloacetate, we just put it through a dehydrogenase reaction, which means we just remove an NADH or the hydrogen from malate, and eventually turns into oxaloacetate. So if we need to go the other way, we're going to throw a hydrogen on to oxaloacetate right here. So pyruvate gets acted upon by pyruvate carboxylase, gets an additional carbon thrown on. We form oxaloacetate, and now we want to get oxaloacetate out of the mitochondria and out in the surrounding system so we can go upstream and eventually go through gluconeogenesis. So what we're going to do is throw on a hydrogen onto oxaloacetate, which will kind of reduce it back into malate. Malate is able to cross the mitochondrial membrane, so the malate is going to cross the mitochondrial membrane here, and now we have malate on the outside of the cell. And all we're going to do again is just remove a hydrogen like I just described up here. So malate is going to be acted upon by another dehydrogenase, malate dehydrogenase most likely, and we're going to produce an NADH right here. We're also going to produce some energy through a redox reaction, but now we are able to have oxaloacetate on the outside of the cell. And if you guys recall earlier, I talked about, I think it was phenylalanine pyruvate or PEP. Oxaloacetate is going to be acted on by GTP, which is basically the same as ATP. It's another source of energy. And this enzyme right here, PEPCK, is just going to be the enzyme that helps convert oxaloacetate into PEP. So once we get to PEP here, we can basically go through gluconeogenesis. And PEP will turn back into 2-phosphoglycerate, 3-phosphoglycerate, go all the way back up to 1,3-biphosphoglycerate into glyceraldehyde 3P, and then we'll eventually get back to fructose 1,6-biphosphate. We're just working right back up the glycolytic pathway. And again, instead of making energy, we are consuming energy because we're going backwards in form. And we'll get into this system in a second. Right down below, where's the back button right there? Okay. But again, if we get to PEP and let's say our body doesn't need gluconeogenesis, we can just remove a phosphate group from it and just oxidize PEP back into pyruvate right here. Normally how we did it in the glycolytic pathway. So PEP, pyruvate, lactate, end stages of glycolysis right here, and again, produce 1 ATP. And that's just what's going on right there. And we can convert it back into pyruvate, and pyruvate can go do whatever it wants to do if we do not need it for gluconeogenesis. But if we do need PEP for gluconeogenesis, none of this stuff is going to happen right there. Yeah. And what I also want you guys to know is we can see that the TCA cycle right here is also involved in gluconeogenesis because we will eventually make oxaloacetate, which will again will go through the malate shuttle and go all the way back up into fructose 6,1-biphosphate, which is just again moving back up the chain because we want to get glucose back into our blood. Honestly, and again, happens twice because we need 2 PEP to produce 1 glucose, because 1 glucose will produce 2 of all these products, so we need 2 of each product to go back and create 1 glucose. All right. So all this previous stuff I've tutored at least once or twice already this week, but this regulation of gluconeogenesis, this is going to be my first time kind of running through it, so it might be a little hiccupy, but we're going to go through it. So there's a lot of confusing stuff here. And I remember the first time going through this, I was completely lost, and I just had to reread it a little bit just now, just to work out the initial confusion I just had. But it's honestly one I'm hoping if I can break it down clean enough, it won't be an issue for you guys. So basically, regulation of gluconeogenesis is going to be due to this molecule right here, which is going to be fructose 2,6-biphosphate. And fructose 2,6-biphosphate is a regulatory molecule that regulates between either going through glycolysis or gluconeogenesis. So if you remember, glycolysis is the breakdown of glucose, and gluconeogenesis is the recreation of glucose. And this molecule is going to help regulate what our body needs to do. It's going to say, hey, you either do gluconeogenesis or you do glycolysis. So, all right, let's get into it. So fructose 6P is going to be one of the main molecules that's either created or turned into fructose 6-biphosphate. As we can see, if fructose 6P is converted into fructose 2,6-biphosphate, the main enzyme that's going to make that happen is going to be PFK2. And then if we need to turn fructose 2,6-biphosphate back into fructose 6P, the main enzyme that is going to do that is going to be fructose 2,6-biphosphatase. Now, what we need to look right here is the more fructose 2,6-biphosphate that we have, that means our body is going to want to be going through glycolysis a lot more. That means it's going to have a higher initiative to go through glycolysis because we are having abundance of storage of these molecules, and we're just kind of going to be going through glycolysis. Now, if we have less fructose 2,6-biphosphate, which means we are converting it back into fructose 6P, that means our body is going to be in a state of gluconeogenesis, because fructose 6P, if we convert fructose 2,6-biphosphate back into fructose 6P, that means we are putting it back into gluconeogenesis, and fructose 6P is going to shoot back up here to be made into glucose to be put back into our blood. But if we're taking fructose 6P and turning it into fructose 2,6-biphosphate instead, that means we are trying to break down and trying to get it out. I'm trying to figure out the best way to explain this. I am kind of stumbling over my words here. It means we're just trying to get it out. And I really want you guys just to know, if we have a high abundance of fructose 2,6-biphosphate, that means we are going to be in a state of glycolysis. Now, when we have a high abundance of fructose 2,6-biphosphate, that is going to be a positive modulator for PFK1. And if you guys remember, PFK1 is the enzyme that controls the conversion of fructose 6P to fructose 1,6-biphosphate. So if we have a high abundance of fructose 2,6P, that means we are going to be activating PFK1 and initiating the conversion of fructose 6P into fructose 1,6-biphosphate. And on top of that too, if we have a high abundance of fructose 2,6-biphosphate, it is going to be a negative modulator for fructose 1,6-biphosphatase. And fructose 1,6-biphosphatase is basically the enzyme that converts fructose 1,6-biphosphate back into fructose 6P, which again is a characteristic of gluconeogenesis, because if we remember, we are working back upstream. So if we have a lot of this, we are activating PFK1, which is initiating glycolysis, and we are also inhibiting fructose 1,6-biphosphate, which is preventing gluconeogenesis. Now, if we do not have a lot of fructose 2,6-biphosphate here, that means PFK is being inhibited and it is being shut off. So this conversion of fructose 6P to fructose 1,6-biphosphate is not occurring because the low amount of fructose 2,6-biphosphate is inhibiting this enzyme. And if we have a low amount of fructose 2,6-biphosphate, that means we are going to be activating fructose 1,6-biphosphatase, which means that this enzyme is going to be firing and converting fructose 1,6-biphosphate into fructose 6P to continue going upstream into glucose 6 phosphate, yeah, G6P, whatever, which will eventually be turned back into glucose. Cool. Yeah, so the less fructose 2,6-biphosphate we have is going to be initiating gluconeogenesis, and this is just due to low blood sugar. And as I've repeated in numerous videos, gluconeogenesis is just trying to get glucose back into our blood to raise our blood sugar levels. Cool. All right, that was a lot of talking right there. I hope I broke it down pretty good, honestly. I was just kind of repeating myself a lot, trying to get these point across, and I hope it wasn't too confusing. But again, as I've said before, if you guys have any questions, please never be afraid to reach out, shoot me a text, give me a call. I know I'm putting this out pretty close to your guys' exam date, but I'm always happy to help and always ready to clear some confusion. So if you guys have any questions, please feel free to reach out and well, have a happy Saturday. Go enjoy the heat advisory, the heat wave, catch some rays. All right, see you folks.